Thank you very much and good morning. I'm going to spend about 25 to 30 minutes just giving you an overall explanation of how immunotherapy is currently working, and then we'll get to a panel discussion. So to start off with, I think you heard a little bit that immunotherapy didn't just happen in the past 10 years. In fact, it has a long history, and that history began with William Coley back in the 1890s. You heard about him, a surgeon who made an important observation. He realized that some patients who got infection, who had cancer, their cancers would regress. He subsequently decided to inject actual tumors with infectious agents. And in some cases, such as in sarcomas, he actually saw regressions. So that was the beginning. Paul Ehrlich, soon after, was very well known as well for coming up with the term magic bullet. I don't know if you've heard that term. But when he talked about the magic bullet, what he was really talking about were antibodies that could fight threats. And of course, in his day, the biggest threat was infection. But now we know that cancer as well is a threat. And in fact, it's because we have so many good treatments and preventions for infection that people live long enough to get cancer. I don't know if you've thought about it in that way, but it's really an interesting way to think about this. And so now our next, of course, big challenge is to get rid of cancer, and that's why we're here today. So, and then you have Dr. Lloyd Old, who many of us in the field knew and looked to him as really ushering in the more modern era of vaccines. He had shown that BCG, which is basically a tuberculosis extract, he was able to use that in mice to prevent cancer from developing. Well, this is important because actually BCG now is used to treat superficial bladder cancer. So he really developed one of the early immunotherapies in this field. And so I also want to point out, so until recently, most of treatment was done with, as you know, surgery, chemotherapy, radiation therapy. And this is just showing you sort of a survival curve and showing you how the standard therapies and the immunotherapies are different. So we're seeing an increase in survival benefits, significant increase over the first 3 years of treatment. And then the curves, of course, unfortunately reach a plateau, a low plateau, but still a big difference. But we realize that although we've made some good progress, and we've increased survival, we still have a lot of room to go. And there's a lot of new therapies, and I encourage all of you to learn about them because these new therapies are in testing. And it used to be 20 years ago, you go on an experimental therapy and the chances of surviving and doing well with it were not very good. But think about these patients who went on the first therapies of immunotherapy in the past decade. They're still alive and doing well. So I think experimental therapies have a different connotation now than it did even 20 years ago. So what about the immune system? So this is the immune system at a glance. And as you know, it's our natural defense against infection. But it's also really, when you think about it, you break it down, it's a collaborative network of cells, organs, and molecules. And they're all interacting to react to threats. The threat being either infection or, in the case that we're talking about, cancer. And these cells, the immune system develops in all different organs within the body, and they're all very important, and they all have a different role in developing the immune system. And there's both the adaptive immune system as well as the first defense immune system. So the cells of the immune system really consist of these different cells. And so you have your first responders, and your first responders are going to be the first responders in infection. It's going to be neutrophils, monocytes, natural killer cells. They can almost immediately see a threat. Okay, so they can see bacteria, virus, they come in right away, and often these cells have the ability to either basically eat the threat, so in the case of infection, or they can actually kill the threat initially with acute types of mechanisms. But it's not really a cancer-specific mechanism yet. We need to go to the adaptive immune system to really get to the specific, cancer-specific immune system. And that's done by the first responders, such as macrophages, as well as specialized cells called dendritic cells, where they take up the threat, whether it's an infection or whether it's against the cancer cells, and then they process that threat in a way that they can present it to two arms of the immune system, the B cells and the T cells, which can specifically react against the threat. So we'll go through that. So this is your dendritic cell, and your dendritic cell can engulf a tumor cell, okay? Under the right circumstances, they have to be alerted to the tumor cell being there, but they can see differences between the normal cell and the tumor cell, and that's why the immune system is so powerful. And so then what it does is it breaks down the cancer cells into proteins that it can now actually present on its surface on a molecule called the MHC1 molecule, major histocompatibility class 1 molecule. So no need to know that. All you need to know is that it has to break down the cancer to a form that can be presented. And why is that? Well, because these specialized immune cells, the T cells in particular, have the ability through that receptor I'm showing you there with the arrow to see that threat, but only when it's bound to the MHC molecule. So the T cells are patrolling. They're coming from the lymph nodes and the spleen and the blood, and they're patrolling to look for threats. That's what the immune system does. And the threat can be infection, but it can also be cancer. The problem is, what happens when it sees it is it does have the ability to kill the tumor cell at this point because it's being activated by the antigen-presenting cell. But it doesn't want to kill the antigen-presenting cell. It really wants to kill the cancer cell. So now it has to go on a seek and destroy mission to find the tumor cell. And the cancer cell is presenting the same threat. So that's what's nice about this. The dendritic cell takes up the cancer cell, gets the right proteins to educate the immune cell. The immune cell now goes and seeks the cancer cells all over the body. But under some circumstances, it's eliminated. But as you know, you've heard, we can also, unfortunately, the immune system not only is there to inhibit infection, but when infection is over, there are mechanisms to turn off the immune system. Unfortunately, the cancer is very smart. It knows how to turn off the immune system, and that's what it does. So it upregulates a second molecule called PD-L1. And there's PD-1, which it can interact with on the immune cell. So this is where immunotherapy has made great strides, right? Because while PD-L1 is acting as a brake on the T cell, on the T cell to prevent it from killing the tumor cell, what happens is we can, and what happens when that occurs, the T cell goes into exhaustion. It no longer can do its job. But we come in with a checkpoint. It's an antibody that blocks. It just blocks either this signal or that signal. So the brake's off. No brake. Okay? So that's what we're doing with immunotherapy. You remove the brake, and now you have an activated killer T cell. So that's how the current immunotherapy called immune checkpoints are working. And they're very effective in some cancers. And so pathway blocked, cancer cell eliminated. And it's amazing because, as you heard in the video, when patients respond, they respond pretty quickly. And the tumors can actually melt away, and patients can have metastatic disease in some cases and end up in a total response, complete response, and go on for years. And those are the best cases. Of course, there's the intermediate cases, but those are the best cases. That's what we're trying to achieve for all patients. So another type of therapy is called adoptive immunotherapy. So you may have heard about this too, because CAR T cells are now approved for a number of indications, particularly for childhood leukemias, which is typically deadly. So we're able now to have a good effect for some patients, particularly young ones. So talking about adoptive therapy, it also has a long history. Okay, it's been around since the 70s or 80s, where we've been trying to isolate patients' own immune system or immune cells. So we isolate it from, let's say, like taking blood, a leukapheresis. I don't know if you've heard of that, but it's the same thing. You could take out the blood cells, and now you can activate them in a culture dish, get them activated, and then you can give them back. And when you're activating them, you're basically educating them to react against the cancer. Okay? So, and this works pretty well for cancers like melanoma, because melanoma is a cancer that it's a UV-induced cancer, and what that means is that it makes a lot of those proteins that the immune system can see, because the UV is basically a carcinogen, causes a lot of DNA damage, and then you get a lot of pieces of proteins that the immune system can react against. So now we've moved from this adoptive therapy, which works for some patients, in particular melanoma, to what we now call T-cell receptor engineering therapies, and those are the CAR T-cells. And so what we're doing is we're still taking a patient's own immune cells out, but now we can engineer them. We can engineer them with a protein that can seek the tumor protein, an antibody that can seek the tumor protein. And we can also engineer them to activate upon interacting with the tumor. So that's what's really cool about these CAR T cells. So you have your T cell, and what we do is we put in this receptor here. Now this isn't going to see a MHC molecule with the piece of tumor. Instead, it's going to see a whole protein on the tumor. And that's the difference. And so they enable what we call an MHC independent targeting. So it's a second way. So you can see where our field now is coming up with multiple different ways to use the immune system to trick the cancer and get rid of the cancer. And so the CAR T cell interacts with a whole protein on the tumor, but that's not enough, of course. So it's also engineered to be able to become activated itself and kill the tumor. So the T cell interacts, and then there's a mechanism that allows it to say, hey, I got to kill the tumor. And so what we can do with these CAR T cells is we can engineer them in many different ways. So we could start out with a lymphoma, okay, and that's what it was originally, a leukemia or lymphoma. That's what it originally was approved for. But we can engineer it to express a protein on a breast cancer cell or on a colon cancer cell. Now, it's a little tricky because we have to find the right proteins that are different from the normal cell, but that is where the field is evolving. And then we can also figure out, are there other ways to better activate those cells? And so we can continue to engineer these cells. So the engineering component of what we do has really advanced over the past 10 years. So another interesting approach is called the oncolytic virus immunotherapy, and that's also been approved for melanoma. And so viruses can alter our cells' DNA because it inserts their own genetic material, right? And viruses are one of the danger signals to the immune system, the threat. The immune system loves to react to viruses. And so, as it says, you have impaired defenses that make the tumor cells more susceptible to infection, okay? So it's easy to get this infection into the tumor cells. And after injection, the virus causes the tumor cell to basically burst and release all of those proteins that the dendritic cell can come in and now take up and then activate a B cell or a T cell. So the immune cells uptake and present those tumor antigens, and then they stimulate the B or the T cell, which is the adaptive immune response. And so we can actually reprogram these oncolytic viruses too. We don't have to just give a virus in its natural form. In fact, you don't want to do that because you don't want to cause a viral syndrome in the patient. And so what we do is we remove the disease-causing genes, right? And we're able to insert immune-stimulating genes. So let's make it even more powerful and say, hey, to the dendritic cell, I'm here, come get me. Come present me to the immune system. And so the final thing is the cancer vaccine. So remember we started back in the 1890s with William Coley. Well, we're back to cancer vaccines. So immunotherapy works very well in some cancers because those cancers naturally present those proteins or those antigens. But sometimes we have to first tweak the immune system and figure out what the actual cancer cell is expressing and then give a vaccine, not in the sense of preventing right now, but to activate those T cells and B cells, to tell the dendritic cell, react against these fewer number of proteins in the cancer, but they're there, and you need to see them. And so that's what tumor vaccines do. So we take the tumor antigen, and there are many ways to give the tumor antigen, and often it's just an injection under the skin as if you're getting a vaccine against the flu. And then the dendritic cell will take it up because within that vaccine that we're giving, we also give an alerting signal. Okay, so we mix the antigen with an alerting signal. And so the dendritic cell knows to come and take it up. It presents it on its MHC molecule. The T cell gets alerted, it gets educated, and it becomes activated. And so many of the studies that we're currently doing with this is we're not only just giving vaccines, because with lots of cancer on board, it's very hard for a vaccine to do its job. And remember, what happens is often the tumor cell's smart, the activated T cell gets in, and then it puts up those brakes. So we have to give the vaccine with the brakes. So you'll see that there are clinical trials that are ongoing that are having potential good results where we're combining the two now. And so where's the future going? So the future is really going from saying, well, colon cancers express these number of proteins, all colon cancer patients are going to get the same protein vaccines, to saying, hey, each patient is individual. Their cancers, although they may start in the same way, they're developing differently and may have different proteins. And so now we can just take a small biopsy from a patient's tumor. Within a week, we can sequence all of those proteins. Again, this is something we couldn't even do 10 years ago this quickly. So you could see where the technology has really evolved. And then we could determine which proteins the cancer cells are expressing out of the ones we sequence, because we're going to sequence a lot of proteins. And then we can make the vaccine that's personalized to the patient. And that's what you're seeing here. And so it takes a biopsy, it takes sequencing, which is about a week, maybe 2 weeks at most. And then we have specialized scientists, bioinformatics individuals, so they're data scientists. We're bringing in lots of data scientists now, people trained in mathematics and engineering, where they develop these programs where they can take all of this information, and it's a lot of information, and that's one of our challenges now, being able to understand all the information we're getting from cancers as we study them. And they could determine which of the proteins we've sequenced are important to a patient. And then we can say, we could take immune cells from the patient by just taking a blood sample and see which ones the immune system actually sees based on the predictions. And then we can make a peptide vaccine, and then we can vial it, of course, and we can give it back to the patient. So this is already starting to happen. I can't tell you that we've had a lot of progress yet in it, but it's definitely happening, and you're going to be seeing more of this kind of therapy, as well as when we take that biopsy, we can figure out what are the different breaks that are going up once you give a vaccine. So we may be able to personalize the type of immunotherapies we give, both the vaccine and the immune checkpoints. And also, understanding these proteins helps us develop new CAR T cells, right? CAR T cells that will target a patient-specific tumor. So what are the challenges then? So the challenges are discovering and validating new biomarkers to help us predict which patients will respond to which therapies. It's still a challenge. We have a couple of biomarkers. They're not perfect. They're pretty good. But they only work in those cancers right now where we're seeing good responses, which makes sense. So a lot of research is on trying to figure out the cancers that aren't responding. Can we predict biomarkers that will help us figure out why they're not responding and help us make the therapies better? We also need to determine the best way to combine our immunotherapies. I told you we can do this, but that doesn't mean it's easy. So we're still learning how best to do that. And to be honest with you, we could do preclinical studies and other types of models, but the clinical trials are really what helps us learn. And so by patients being a part of the team, it really helps us make the therapies better. And we're also, the important point about immunotherapy, we all know that it has toxicities, as does any kind of drug. And these happen to be a different kind of toxicity than chemotherapy. It's called autoimmunity, where sometimes when we activate the cells to fight the cancer, we're also activating cells that can unfortunately hurt normal cells. And so we're now learning how to decouple those side effects from the benefits of immunotherapy. And that's definitely an ongoing area of research. So why have most responses been modest, and why are some cancers refractory to immunotherapy? So what you're looking at here is that T cell, that immune cell, and that dendritic cell or tumor cell. And what happens, that what we've learned over the past probably 20 years, is that it's not just one signal that the T cell gets. In fact, again, remember, a T cell or a B cell really has to be regulated. It has to be regulated because they come in, they see the threat, they get rid of the threat, and then something has to turn it off. That's natural. It's a natural process, right? So a T cell has both activating and down-regulatory, so brakes and accelerators on it, and it interacts with either the antigen-presenting cell, such as the dendritic cell, or the tumor cell. And so what we're learning is which of those accelerators and brakes are most important to modulate for each patient. And that's where the research is going. And so for the patients who respond, we know the answer. Okay, at least we know one answer. But for those who don't respond, which is the majority right now still of patients, we have to figure out what other brakes or accelerators. And more than likely, it's going to be combining a number of them. So cancers are complex, and so we have to better understand them, and we're likely going to have to give combinations. So, and then the other problem is that cancers secrete chemicals to turn off the immune system. So cancers, unfortunately, have multiple ways to evade recognition by the immune system. And another way is that they can just express chemicals that turn off specifically T cells and B cells. So why have most responses been modest, and why are some cancers refractory to immunotherapy? So again, cancers can recruit suppressive cells to activate and block the immune response. So that's a third reason, okay? And so what you're looking at here, we can stain for them now. We could take a biopsy. We can stain for all different kinds of cells, both the ones that are going to kill the cancer and the ones that are going to suppress the immune system. So again, immune cells, they're not all good, okay? They can be taught by the cancer cells to prevent the T cells or B cells from seeing it. In particular, these cells here, the macrophages, CD68, the yellow cells. So often when I look at a cancer that doesn't naturally respond, I'm going to see a lot of those cells. But we're learning what the signals are on those cells so we can, again, try to prevent them from suppressing the immune response. So research is finding new ways to meet these challenges through the identification of additional cancer targets. So again, I told you about how we're able to now sequence tumors. We can identify new targets through integrating multiple targeted therapies, again, bringing different types of therapies together. And sometimes it's going to be another kind of targeted therapy, not an immunotherapy, that can hit a gene that's upregulated in a cancer and then combining it with immunotherapy. So we're thinking about lots of different ways of trying to stop cancer. And most importantly, and I truly believe this, and I tell my patients this, that patients are part of the team. They're helping us to really figure this out. And we are so blessed to have all of you interested in our clinical trials and willing to take part in them. And the good news is now I can honestly say that there's much more hope with clinical trials than ever before, and I think this is going to continue. This is only going to get better. We really are at a time that's a time like no other in our field, where we are just making rapid progress. Just as an example, over the past 5 years, there's been more than 30 approvals of immunotherapies for 30 different indications. So that's the kind of progress we're making. And we had 30 years of very little progress, and then all of a sudden we're making this rapid progress.